Thank you all for coming. It's my pleasure to introduce today's speaker, Professor Jeorg Lahann. Dr. Lahann is a professor of chemical engineering, material sciences, engineering, and biomedical engineering here at U. He also the director of Biointerfaces Institute, as well as he's the co-director of the Institute of Functional Interfaces at Karlsruhe Institute of Technology in Germany. So Jeorg is a co-inventor of Greenmark's dental technology. He has co-authors more than 200 papers, more than 40 patents, and he has been selected to review as one of the top 100 young scientists and the participant of 2007 Nanoscale Science Engineering Award. Jeorg has his TV is so big, so I will Jeorg, welcome to Department of Computational Biology and Bioinformatics, Computational Medicine and Bioinformatics. It's all yours, Jeorg. Thank you, Jeorg.
Well, thank you very much for inviting me, and it's in many ways it's a very short and a very long journey for me. Right, the short is took me 10 minutes to get here from NTSC, and the long one is because I think we can learn a lot from each other, because my lab really focuses more on materials and how we can make materials, design, synthesize materials for biomedical applications. And so we'll focus on a couple of examples where we use computational tools as well. But the truth is, a lot of my students, they work really still with hoods and flasks in the lab, so very much like hands-on experimental work. But nevertheless, I think there are a lot of possibilities for us to collaborate, and so in that sense, I'm very excited about being here today. And so this is on, right? Yes. You guys can hear me online too? Okay, good. So before I get started, I'm also part of the Biointerface Institute. I'm very excited about that, and we'll tell you a couple of things about that. And then I just picked 3 vignettes actually for my research. Hopefully, I don't go very much into depth in each of the or in any of those, but hopefully that gives you a feeling about the kind of stuff that's going on in my lab actually. So the Biointerface Institute has been founded 10 years ago. So similar to your department, so we're kind of twins growing up about parallel, I think. So very, very similar. We have faculty from 30 different research or students from 30 different research groups within the institute, faculty from 13 different departments. And the Biointerface Institute is cross-cutting between engineering, the medical school, pharmacy, and the dental school. The couple of aspects I think that make us unique. First of all, the breadth of the faculty and the real focus on collaboration. Obviously, a lot of other places do this too, but I think that's really important to us. The way we pick particular clusters and themes to organize our faculty around. Also, the focus on translation. So we're very much interested in bringing our technologies to essentially clinical researchers and ultimately turn them into health benefits. So in many ways, when we started this, the idea was to connect, kind of engineers, physical scientists like me, with more translationally savvy clinical researchers that can impact and guide essentially our research way earlier on. And so this is kind of the premise of the Biointerface Institute. If you have more questions, so as we get to our 10th anniversary, we have a lectureship research symposium coming up. It's on November 10th. So you're all invited very much. We'll give you more information. It's going to be not in the big house, but in one of the side rooms of the big house. So we're not that ambitious. But nevertheless, I think it's going to be a great event. We have a lot of nice speakers lined up already, and you'll hear more from that as we go through. I think the first email actually went out today. So, or ask me afterwards, that's fine too. So coming to my group, so basically working on polymers and surfaces, and I'll give you an example for how we use defined surfaces, engineer very well defined surfaces to do certain things in the biomedical space. We also work on drug delivery systems. Specifically, my group has interest in developing particles that are tailored towards siRNA delivery and plasmid delivery more recently. I'll talk about one example in the context of glioblastoma, a collaboration with Maria Castro in neurosurgery. And then I'll talk a little bit about engineered microtissues as well and technologies that we are developing towards those applications. So starting with the first one, so it's going to be a nanoparticle delivery vignette. So one of the big issues basically in general therapeutic treatments is essentially to open up and enlarge the therapeutic window in many ways, right? So basically particles, nanoparticles potentially can be a tool to change how a drug distributes within the body, how to mitigate some of the side effects, meaning side effects are mostly coming from accumulation of a drug in the liver and in the spleen and some of those organs, and then also to increase the overall dose that goes into the target of interest. In our case, in the context of this will be glioblastoma, brain tumors. There are a lot of particles out there, right? So a lot of things that people have tried because of that premise. So that idea of a golden bullet that essentially finds the magic way into the target organ, that has inspired a lot of scientists. There are polymer particles, there are the gold nanoparticles. There are a number of lipid nanoparticles, obviously for vaccines, very widely used. And my group more recently is really focusing on another building block, which are proteins. So we actually built synthetic nanoparticles from proteins. And so we try to combine essentially the ability of man-made, synthetically engineered particles with that natural ability of the body to respond to proteins. And the other thing we like to do is think about how to develop particles, nanoparticles, microparticles that are specifically suitable for the delivery of multiple drugs. So think about combination therapies. How do you deliver actually 2 drugs to the same organ and orchestrated in a well-defined way? And so our solution or approach to this is to create particles that have multiple compartments. So 2 hemispheres, for instance, maybe 3 compartments, 4 compartments that are designed to sequester a particular drug and release it independent of the other compartment for tailored towards that particular drug. And I'll give you one example where we actually use this now for glioblastoma. Okay, so the idea is really, you take 3 properties, 3 polymer matrices, 3 carrier properties, and you combine them so that each of them within one single particle, so each of them can really play to their maximal strengths, right? And so that's given here, and it's certainly different from essentially just encapsulating some of those matrices into a larger environment, because if I would release from here or here, I would still basically define the release kinetics and the properties through that and that outside compartment here, right? So this is clearly distinct. So how do you make those kind of particles? I mean, nanometer particles, nanometer is very small, right? So this is not something that you can easily make. So this is something that we figured out about 10 years ago or so, and we use a process that is essentially very similar to what you use in the mass spec. So in mass spec, in electro spinning specifically, ESI, you're basically using a dilute jet, you bring it into a vacuum. In this case, we don't need vacuum, but you end up with essentially like a particulate that's simply just a single protein often. In our case, it's a little bit different, but very related. So we use a high electrical field as it is used in mass spec, and then drive a fluid solution that is basically comprised of our protein and maybe a polymer matrix towards a counter electrode. This is an example, and you see here basically a macroscopic needle. You see a little droplet there. Think faucet at home, right? So when you let your faucet, when it drops, you see a little fluid droplet form at the end of it. The difference here to the faucet is there is a laminar flow that is coming down that's comprised of 2 independent streams. And the laminar flow is important because then we don't have convective mixing. So even though these fluids are miscible, they still stay separate, right? So that's nice. Now we use electrical fields. I can move a little bit. And drive this against, essentially, drive a jet against a counter electrode. So you see the jet coming out here. And really the clue is, the magic is that jet still remembers it was made from that laminar flow. So in the end, we basically eject, we decrease the diameter by multiple orders of magnitude. So we basically increase the surface area. We get instantaneous precipitation. We end up with solid particles. They look like this.
How do you keep those parallel flows?
So it's just because of the inertia of the system and the velocity that you see. Thermodynamically, obviously, you would want, or you would expect that they form core shell with the lower energy outside. But because of the rapid process, we keep them separate, as I will show you in a second. So we collect solid particles. They look like this, very much like Electro Spinning. But then if you do microscopy, you can see they maintain this compartmentalization. So that's cool, because all I have to do now is I'll say, OK, I'll put track 1 here and track 2 here. I mix a little bit of polymer in there. It's fine that this is completely dilute, because through my precipitation, I end up with a solid matrix that basically sequesters my track, right? So in the context of what I'm going to be talking about, this could be a polymer with a siRNA, for instance. And this could be a small molecule drug like paclitaxel. And you can combine them and create particles that basically deliver both, but with different kinetics, because the kinetics I can control by the choice of the matrix and the drug. So this is how they look. So more than, we do flow cytometry with them if they're bigger. So more than 95, 98% are bicompartmental. This is even without optimization. Sometimes we fraction them out to better clean them off and make even better distributions. So they look very nicely. We can load them with a number of different drugs. Here's some typical drugs that we've been working with. So this is actually a very general concept, because it's so simple, right? The beauty is simplicity. Okay, so coming back, I told you leading up to this, we actually now move from the synthetic polymer, such as the polyester that I used here, which is an FDA-approved polyester. So that's a nice thing. But we now use proteins as building blocks. So it's a little bit like Apraxin, but the difference between Apraxin and something like this is that Apraxin is only physically cross-linked. So the moment this gets into the blood, it falls apart. Whereas here, we chemically stabilize it. So the particles themselves are much more stable. But then when they get to the liver, you still have proteolytic cleavage of the protein part of the particle, and you have very rapid liver clearance, right? And so you can see, for instance, that the particles are well distributed, about 100, 150 nanometer in size. The size distribution, even in a aqueous environment, is well defined. It's a little bit bigger because the protein swells. That's actually nice, because we also see a pH dependence, which allows us with coming out of the endosome. If we go into the cytoplasm, we have to come out of the endosome, which has a lower pH. That's a very nice system. So those are great particles. They're stable over 10 days. So we're looking at the diameter over a long time. So they're stable, which is good. This doesn't work anymore, but that's okay. And we can make them from a lot of different proteins. So we just used a few. We used transferrin, we used insulin, lysozyme, albumin, obviously, I showed you already. But the nice thing is you could bring potentially the ability to have access to a lot of proteins to bear, and also the ability of proteins to home into specific organs. So tropism is something that you could exploit potentially for those particles. We haven't done this as much, but I think there's a real opportunity here. And then obviously the ratio of this synthetic component and the protein will also set things like mesh sizes, release kinetics, stability, all of those things. So it's a really engineered system, which is nice. So let's talk about tissue homing a little bit. And this is a study that actually is a collaborative paper, but it's really came out of UPenn. So Jake Brenner and Vladimir Muskantos are the corresponding authors. Our group is just a co-author on this. But I still think it's worthwhile to mention, because what they did is they took about 25 nanoparticles or something like this, right? So a very large library of particles, several of hours as well. And they asked a simple question. Is the biodistribution of those particles different in a disease model, in a disease context, than in a healthy context? So the particular system they looked at was sepsis. So they took a sepsis model and then compared the biodistribution of particles relative to a healthy mouse model. And so some of the tissues were, some of the particles were tissue selective, specifically to the lung. As you can see, so naive is the healthy. This is a sepsis model here, and you see huge differences. But for others, such as viruses or liposomes, you don't see a big difference. So that's interesting because there seems to be something about like the emergence of tropism. Now, this doesn't seem to be exactly protein specific. All of what we do here is albumin nanoparticles. Okay, there's bovine albumin versus human albumin, but overall, it's all the very similar compositions, right? So how about if we look at different protein nanoparticles? And so we did. So they also looked at hemoglobin, transferrin, all the particles that we made for them in the study. And then they look at the ratio of lung to liver. And you see very nice differences here. And some of them have very high lung to liver ratios in the case of sepsis. So what that suggests is there might be a potential to use this type of particles to not only home into particular tissues, but also do this in the context of a particular disease, which I think is very interesting. Now, this is one example. Obviously, there are many other diseases. I don't know if sepsis and lungs is the very best example, but we hope we could do things like that also for the CNS, for brain tumors, and so on. Okay, so let's stay with glioblastoma a little bit more. So this is work with Maria Castro, Petro Lowenstein, and their groups. And so in this case, we make protein nanoparticles, so very much human serum albumin, the same concept, a little bit different cartoon, but like essentially the same type of particle. But now we also include an siRNA that targets STAT3. So small interfering RNA for STAT3. And so basically, we complex that to a PI so to prevent it from diffusing too fast out. We also add a small peptide IRGD, which gives us, which is a transcytotic peptide. Okay, Rushlati has published on this a long time ago. And so we use this to basically induce transcytosis into the glioblastoma. That's the idea. Okay, so we can show with ultra high resolution microscopy that we have a co-location of the protein, which we can easily fluorescent stain, and siRNA, which is also stained in red. So you see nice co-location, homogeneous distribution, and you get sustained release over a couple of days. So that's nice. And that's not so easy with small particles. Right? I mean, think about that, what the diffusion time of a small molecule over 100 nanometers is. What do you think if it's rediffusing? Probably seconds, right? So you do have to have processes to sequester and control that release. So it works with GFP siRNA. So that was our first test case. The paper also uses STAT3, obviously in vitro, but I just want to show it looks nicely and it works similar to lipofectamine, maybe in this particular case, even a little bit better. And then Maria's group basically took those particles and injected them into mice that had the intracranial brain tumor. So these are GL26 cells. The dose is about 10 to the 11 particles, single injection, and we saw statistically increased survival. So typically, this is an aggressive tumor. So typically, the mice die about 28 days into the tumor. And we saw, a sizable, but, maybe not super exciting extension in survival, but it's statistically significant. Then what we did is we basically followed that up with multiple injections. So it's a cascade of these injections over 25 days or 20 days, actually. And now we see even higher benefit from the nanoparticles, much better than the free STAT3, where we see pretty much nothing, which is here or a little bit. And obviously the control or the particles themselves, the mice die very, very rapidly. Now, STAT3 is a hub, but even in vitro, when we do our IC50 measurements, it's relatively non-lethal, right? So it's not a very lethal pathway, but combining it with a second method seemed to be very interesting. And most importantly, the first line of treatment for glioblastoma is ionized radiation. So the logical thing to do is to combine this now with ionized radiation. And that's what I'm going to show you next. So now we're basically combining ionized radiation, multiple treatments, with our particle, the same particle regimen that I showed you. And now we're basically seeing in or 7 out of 8 mice, we see full survival. So I learned the mouse is a survivor after 90 days. So those guys survive for 90 days, which is great. We see P*3 and STAT3 levels that are indicative of activity of the STAT3i. We also in this paper showed that the particles are going into the glioblastoma, into the brain tumors. And also when we do histology afterwards, here's basically histology of a tumor in a mouse that was not treated with a mouse that was treated. And we see very nice effects. And we also see influx of immune cells. So increased influx of neutrophils, maybe macrophages. So that's also very interesting. So this suggests there is a secondary effect here. And when we then also re-challenge those mice with a second tumor on the other side, it seems like that there's no tumor formation afterwards. So it seems really that there is an immune memory effect at play here as well. Yeah. So we siRNA. So we block the STAT3 activity.
So how is this targeted only for cancer cells? So what happens with normal?
No, so it targets any cell, and most importantly the liver, right? So because it goes into the liver next. So that was what I was just going to show you.
Okay.
So how does the liver look for those particles, for those cells that were treated with those particles? And we do get relatively quick clearance of the particles from the liver because of the proteolytic activity and the cleavage of the particles. So I don't have an exact answer to your question, but we don't see, and this is done by a pathologist, we don't see gross hemotoxicity in any of those samples. So there's no indication there is a very strong toxic effect from this. Now it has probably to do with STAT3 potentially as well as a target, but we don't see that in this particular case. Okay, so they can be Janus, they can be bicompartmental. So let's see how that would work with bicompartmental particles. So now I'm going to take my siRNA delivery system here and combine it with paclitaxel. And so for paclitaxel, the way we do this is we use high pressure homogenization to bind it to the albumin, because again, I told you about the seconds of nanometer over nanometer delivery times. So we really want to make sure that we don't release too fast. And then combine that, put that in one compartment or one fluid. The other fluid flow is the same that I showed you. We make particles that look pretty much the same. We're still kind of statistically tweaking out how different they are. Beautiful size distributions. And these are ultra high resolution microscopy images that show that they're indeed bicompartmental. And look at just one thing, look at the PDI of those particles. So if you work with lipid nanoparticles, we're very much in that regime here with those particles as well. Okay, so they release, the drugs come out separately. This is siRNA and PTX delivery, so we can control that. In this case, basically these are the release kinetics. And then we're looking in vitro at DAMs. We're looking at markers of immunological death. So again, V is Maria Castro's lab and Pedro Levenstein's lab. And we see basically in all cases that the markers of immunological death are upregulated in our specific groups here, especially if we combine it with ionized radiation.
Also, can you deliver like modified mRNA too, the same technology?
So we have mostly worked with, so the new thing that I'm not going to be talking about today is plasmids. So we've done some work with plasmids. I think I don't see a reason why we couldn't do mRNA too, right? We just haven't spent as much work on that. So at that point, these are a couple of points, I guess. Most importantly, it's a platform, SPNP, so synthetic protein nanoparticles is a platform that is leveraging a number of different proteins. Even though I only spoke about one, there's possibilities to think about others, proteins that can actually be targeting as well. So I hope that there's some tropism here or path towards. We can make very small particles, 100 nanometer, even below 100 nanometer in some cases. We see good GBM targeting and even therapeutic results in some of them, including cancer survivors. And we are now working on bicompartmental particles just to hit the cancer on multiple pathways at the same time, which I believe is a critical thing in those systems. So what I'm going to do now is I'm going to switch to a different part of my lab, but I just wanted to give you an overview on the particles. And if you have any questions, I mentioned plasmid delivery, for instance, something we're doing now. And we have a good system, a generation 2 type of particle. We can talk about that certainly in the future. So here, the in vitro, what I call the in vitro dilemma is basically you would like to have something that is usable, high throughput, doesn't require an art essentially, but then also you want to be relevant. And so, yeah, let's do everything in humans. Wait, that's not possible, unfortunately. So then we can do animal models. Now that's great because they're really complex, but already way different from humans. And so there are issues here as well. And obviously, a Petri dish is a great solution for a lot of things, but not for everything, right? So there is this space in between, which are 3D in vitro systems. And you see gel encapsulation, fibrillar systems, and organoid structures, and many other things. There's a whole literature field, a whole research field that's basically working in that space. So that's kind of what we are interested in as engineers. How can we develop those systems differently and maybe even in some cases better? So what we need is some kind of a scaffold that ideally is defined and very porous, because a lot of stuff, at least we started working on, has porosity, but the pore size is not conducive for cells to actually infiltrate and become 3D. You need cells. And then actually what's not shown here, but is even more important, is the cells have to interact with the scaffolds through proteins. And if you ever looked at the decellularized matrix of a cell that the cell deposits, that's a fibrillar system. It's not your globular fibronectin that you just smear into your Petri dish. So there's an issue there as well. So we're working on two things: the scaffold architecture, making a scalable, easily achievable system with various geometries, and then how do we get proteins into the system? So I talked in the beginning, as I talked about mass spec and electro spinning. If I would have given the scaffold talk, I would have called this electro spinning, right? And probably some of you have heard about electro spinning. It's the same process, only now I'm not going through what's called Rayleigh instabilities. So I keep my thread intact and I make fibers. Perfect. So now I typically, if you do electro spinning, you get this kind of membranes. If you ever done tissue engineering, a lot of people use them with PLGA. They're used in water separation membranes, a very common system. But they always look like these random non-woven structures. And there's a simple physics, right? So if you can see that here, but I guess let's do it here. So if you chat down, the first chat, oh no, it's turned off, but it's going to be straight, completely straight, because that's the shortest distance between your charged needle and the counter electrode. But then when you deposit a little bit of material, because you have such high electrical fields, we talk about kilovolts here, you induce a little bit of charge on the surface. The jet comes down, has the same charge. Now it doesn't want to go there anymore. It moves over, right? So you basically have that elongation, and you go all the way over until inertia pulls you back. So you get what's called a whipping instability. And then depending on where your substrate is, you get this random deposition. So you get those non-wovens. They're actually very simple. So we thought about this. And then, so this is the cartoon to it. I explained this already. And then we thought, well, this is not good for us, because if cells sit on this, this is not 3D. They're still sitting on 2D because the pores are way too small. So we want something like this. So how do you get from something on the left where you have nonwovens with the same material, the same process, same everything? Because this is great. This is scalable. There are a lot of good things with it. It's applicable to a lot of different materials. But you want to have this kind of design control, right? And so all you have to do is when the jet comes down, you basically engineer secondary electrical fields, we call them electromagnetic lenses, to focus that jet and turn the charge hill into a well. And so we basically, this is a cartoon of what we use. Basically, we use a secondary lens. We focus this, and once you focus, you can move a table underneath it that is computer controlled, very simple. And by moving by computer controlling the table, we can basically just write those kind of architectures. So really, these are stacks of fibers. This is one fiber. In matter of fact, this is one fiber that's just set down and follows essentially those lines and is deposited exactly where we want it. So we have a system that is well-defined, gives us very great porosity, right? And also good pore size because cells can actually fit in there and we can make 3D systems. Now, that's good, but I want to minimize the interaction of the cell with the polymer. So I need big pores. So, okay, big pores I have. But now the pore is 100 cell diameter wide. So how do I prevent the cell from falling through and just sitting on the plastic underneath it? And essentially I have gained nothing, right? So there's something that's missing here. I'll tell you how we do this. Okay, we can make tubes too. If you're interested, we'll talk about that. But this is the other thing. We have tessellated structures, but they're not conducive to cell adhesion yet. So we need to do one more thing. And what we have done is we basically explored the roughness, this defined tessellated roughness in those structures to induce, to use shear, like a shear field, to induce fibrillogenesis of a protein, in this case, for instance, fibronectin, over the entire area of our scaffold. So in the end, you get this fibrillar structure of fibronectin that looks very much like the extracellular matrix proteins that a cell would deposit. So that's actually very distinct from just a plasma fibronectin that you coat onto a tissue, a petri dish. Okay, so this is fibronectin, stain for fibronectin. We can basically also confirm the presence of cryptic binding domains in those systems. So this is very much like cell-deposited fibronectin. And the nice thing with this is, because it's shear induced, we can control the shear field relative to our scaffolds, and we can actually induce directionality in those fibers. So by using perpendicular flow to the main direction of our pores, we can then basically align the fibers that we form, the ECM fibers, and we get nice alignment. And in fact, it's actually relevant for cells as well, because that then induces cell organization. We're using this right now in the context of cardiomyocytes mostly, a little bit of cancer cells as well. So there is a little bit of a vignette here where we can actually control the organization of the fibrils themselves through the shear field. And we think there's maybe an effect, sorry, here, so we're looking at proliferation, the different distributions in those cells. There might be an effect, or there seems to be an effect on the cells as well. So these are fibroblasts, obviously, but as I said, we're also looking at cardiomyocytes now, cancer cells a little bit, so this is interesting. In this case, actually, it's not so much the anisotropy that is changing, it's the velocity. So the velocity in the direction is actually the factor that is most influenced by the ECM here. Now, if you take patient-derived samples, this is work with Gary Luca and Max Wischer that we have done, then we can actually expand them on those scaffolds, which I think is really cool because we had very hard time doing this on petri dishes. And we see that basically it seems like we are enriching for like a mesenchymal phenotype in those samples. And so that's something that I think is very interesting if you think about immune cancer vaccine construction, even basic biological studies. So these are the kind of things we're looking at right now. So the question that probably many of you ask now is, how well do the 3D in vitro conditions, our conditions, compare to in vivo biology? Because that's ultimately the holy grail. So it's a very difficult question. And I don't think we have the ultimate answer, but we have done some studies into this. And specifically, we basically looked at the tissue culture polystyrene or cells growing on that, and cells growing in the same cells growing in the same cell type growing in the PDX as a continuum with two extremes. And then we basically compare our scaffolds with matrigel embedded systems and spheroids relative to each other. We basically take a Vario 68. This is a cell that Max and Gary are working with. It's a HER2 negative cell. We take these different culture conditions. We do a drop-seq, a single cell RNA-seq. And then basically we're looking at their transcriptome and compared, and basically we use a machine learning algorithm to basically score how much they are either like plastic or like a PDX. So this is what's shown here. And then basically based on the like a MLP classification of their transcriptome, we find that basically a PDX is obviously very similar to the in vivo environment. TCBS is the most similar to the in vitro environment. And if you classify, obviously we're not like animals, but this is also obviously a mouse system here. These are human systems, but we are seems like there's more complexity in the signaling network of our surface, of the cells on our surface versus the controls. So in summary, what I would like to say is that basically, I think it's important to consider three-dimensional growth systems, as particular fibronectin as the building block of the EMT. We also worked with other ECM components, collagen, laminin, and so on. We see enhanced stemness and elevated levels of chemoresistance. I didn't show that, but we have that in one of our papers already. As cells spend more time in 3D culture environments, they take on more in vivo-like characteristics on our systems, not on plastic, obviously. That's what's interesting to observe. And I think we're still gonna essentially understand further what mechanisms are working here. So this is clearly not an end product. And then the other thing that is cool, what we're doing now is, because it's synthetic, we can plug in other proteins as well. So one of the molecules that we looked at is HA. And so we can dial in HA at different various levels, or high molecular versus low molecular with HA, and then see how that affects, for instance, things like senescence and so on. Okay, so with that, I would like to go to a third minute. How much time? I still have a little bit of time, right? Okay, so perfect. That's all I need. So the last part of my group is basically working on ways to modify existing surface. So we think about implants, we think about maybe even in vitro systems where you want to either create gradients or dial in certain ligands, adhesion molecules combined with growth factors, things like that. Here's a few examples. So we have done some work with Ginobili in the dental space on virus immobilization. So this is local gene therapy, for instance, immobilizing DNA more for biophysical purposes, patterning cells using adhesion peptides. We've done this in little 3D components, microfluidic systems. So this is a very broad interest in my group. And the point is really to develop processes that give you access to very well-defined surfaces. So, and then obviously you want to have very little side effects. So if you look at, for instance, I'm going to talk about this in a second, but some of the early stand coatings that were out there and some of the issues with that, often it was actually the solvent that people used to deposit the polymer that was toxic that actually give you the toxic effects. So often it's the kind of small initiators, plasticizers, solvents that people use to deposit this material that actually makes it toxic. So in our case, we avoid all of this by basically depositing our polymers from the vapor phase. We have a little reactor where we take our monomers, we activate them, and we deposit them from the vapor phase onto our substrate. This is a very conformal coating. It doesn't care about the geometry because there's no solvent, there's no bridging effect. So it gives you very homogeneous deposition. And most importantly, the coatings can be exactly controlled. So we make them 100 nanometer, 80 nanometer thick. And we can dial in the type of chemistry we need. And that chemistry is only poorly picked on what we want to do with that surface afterwards. So if you want to immobilize a peptide, we use something like this. If you want to use light to activate it, we use something like this. If you just want to make it hydrophobic, you can use something like this. If you want to immobilize a thiol, you can use something like this. So we basically have a plug-and-play system where we can dial in whatever kind of our target molecule is. So that works with the viruses, DNA, peptides, proteins, sugars, and so on. Okay, and I mentioned already this works with three-dimensional systems too. This was a while back, but one of my students sacrificed his hair. So we basically patterning on this here with, so there's a photoreactive coating on there, and I'm using stereolithography to pattern that. These are stents, these are microfluidic channels. So we have good control even over three-dimensional systems where maybe spray coating or spin coating is not going to work so well. These are stents. So these are actually not normal metal stents. These are magnesium stents. So they're biodegradable metal stents. And so our coatings can be biodegradable too. So we developed this a few years ago. And so we're basically modifying that to control the release kinetics of the magnesium stents, because in some instances, they release too fast or degrade too fast. And so we can tailor that with our polymer coatings. Okay. Now I'm going to give you one vignette, and the reason why it's there is this is when we use AI. So it's our AI approach, machine learning. So I thought I'd put that in. But even so you do AI, as I will show you, you still need a very well-defined surface to begin with. So that's our CVD process. So we use the CVD process to take a glass slide and put a perfectly defined polyparticle or coating 100 nanometers thick, and it makes the surface exactly hydrophobic. Now we're putting droplets on it. So if you take a needle and put a droplet on it, and that droplet is a protein solution with a little bit of salt. Okay, it's the most simple experiment that you can imagine. And we let it dry over time. And what you see here is this accelerated video of what's happening with the drying. So after this dries, under defined conditions, you do it on a well-defined surface, you get very representative patterns, right? So this proteins actually, or this mixture of protein and salt forms patterns. We found this out actually a while back before people were talking about machine learning and neural networks. And so we saw those kind of, this is a, I think, fibronectin, I believe, protein solution that is dried off. And you see how beautiful they are, right? And interestingly, you know, we do this a few times. If the conditions are defined, the surfaces are defined, they look very similar every time to even to the human eye. So we thought, okay, that's a good starting point. So why don't we do the following? We make a lot of these droplets. And the reason why I think this is a good approach to machine learning is because images are data that are very, so assuming that these are data, so assuming that there is information in this droplet, right? So that's my hypothesis here. This is information rich. If it's information rich, images is something that I can access very fast, much faster than a CD spectrum or an XPS spectrum or something like this. So we make a lot of these droplets. Actually, we have an automated dispensing system that makes them very fast. We have a humidity-controlled chamber where we basically dry them off, and we get these droplets that are very similar to each other every time with very well-defined sizes. Okay, so that's what we do. And we make a lot of them, right? Thousands. And then we ask two questions. So first of all, and then we basically run them through Google's face recognition algorithms that they've developed for the internet. So we use off-the-shelf neural networks and train them against some of those images, and then basically use them to stratify essentially our images. And we do two particular things that we're interested in. We basically take amyloid beta as an example because of its relevance in Alzheimer. So we're basically saying, okay, let's take the amyloid beta peptide and take single point mutations in there. Those are mutations that are also indicative of the different Alzheimer variants. And then we also look at conformational changes that we induce chemically and look at different conformations. And then ask the neural network to tell us what kind of mutation we have in that structure. So I'm talking about a peptide that has 42 amino acids, and I'm changing exactly one, right? This is a very small difference in the structure of the protein. And so the hypothesis is there is a different pattern which the neural network can recognize and then basically tell us which of those we're going to get. Okay, so that's what we do. So this is the example where we take single amino acid mismatches in the amyloid beta. This is the amyloid beta structure. Those are the mismatches that we put in, and those are the images. Some of them are different, obviously, right? You see the difference. But a lot of them look very similar. Then we run them through the machine learning algorithm. And the neural network, we looked at several of them in this paper, but the one that we used here was able to predict or identify, stratify the particular peptide or particular mutation in the peptide with close to 100% accuracy. And I think that's very interesting, just because for me, because of the tiny difference in the protein structure. And I think it would be very difficult to pick that up with any other method.
Sorry, I don't understand the question. You have mutation, single nucleotide.
That's right. I take 9 different peptides. Each of them has 1 different amino acid. So 41 is the same, 1 is different. Not always the same. So whatever is different is shown here, right? So we make hundreds of droplets of each of them, and we feed them into a neural network that Google developed for face recognition. And we ask the question, the ones that we put into the test set, how similar is it to this and that? So we stratify essentially peptides. Okay. And then we do this. And then we can also see obviously where the issues are. These are some of the different networks. We can see how they learn, where they do mistakes. So that's secondary effects that we learn out of this. And then the other thing, which is even more complicated, is now we induce essentially conformational changes. We do this by adding a little bit of a solvent into our water. And that solvent changes essentially the conformational habit or existence of those peptides. We see changes in the helix versus antiparallel versus parallel sheet distribution within those peptides. So to make it easy, here I have a little cartoon. So these are basically 2 extremes. And so the droplets essentially with very high helical contents are on this side, and the one with very low helical content are on this side, and then there are some that are basically in between. So I'm looking essentially at conformational differences in a peptide that otherwise has identical structure. And again, we take images, we run them against our test sets, and we find very high, a little bit lower, but still very high accuracy. You see some of the details here. It's all published too. And so we're very interested in this. Now we're also looking at protein-protein interactions, protein-DNA interactions. We think maybe this is a way to screen interactions as well. So it's a very simple system, right? And one of the issues often with machine learning is how do you get access to a lot of data? Making hundreds and thousands of pictures here is very easy. So with this, it's, I would say, in conclusion, also experimentally extremely simple. These drying droplet patterns are actually indicative of very small structural differences in peptides and proteins. So we use existing machine learning-based techniques. So we don't do our own stuff. We're not smart enough for that, but we just repurpose what's out there. And it works actually pretty well. And then the approach appears to be generalizable, and maybe it could be extended to even protein-protein interactions, different viruses, maybe DNA-protein interactions. So these are the kind of things we're doing next. So with that, I'd like to basically acknowledge the work or the people that did the work. So Jason and then also Eva are the ones that did most of the work on Nahal on the nanoparticle work, the first vignette I talked about. Dylan and Aisha, which is I think not on here, but she left the lab already, did most of the work on the scaffolds. And then Asam is actually the person that did the machine learning stuff with the drying droplets. Disclosures, so green mark, that's right, and then 7 Sense, Gradilis, and the Galactic. So I have interest in those companies. And then collaborators, Maria Castro, Petro Levenstein, Vladimir Mosikantov, and Jake Brenner I mentioned, and then a number of other collaborators as well. And we do some nice work with Indica now too. We're starting to work together, and some funding agencies as well. So thank you very much, and hopefully, I give you a little bit of an overview. If you have any questions, anything about something particular, I understand I went very fast and not very deep, but I thought that's maybe more productive than me hampering on CVD polymerization for an hour. Okay, thank you.
Thank you very much. Your question?
Go ahead. Yeah, use the microphone, I think.
When you were generating the image or the droplets,
Yeah.
did you use the same machine to make all of them? And if so, did you maybe, because like I would imagine that if, you know, a single change in the peptide sequence could cause such a very difference in the droplet formation, that perhaps like tolerances or in like the machines, the variation in tolerances in the machines could also cause similarly very.
You are a non-believer.
What?
You're a non-believer. No, I think you have a good point. So
I'm just saying, like,
yeah.
have you tested to see if like your machine learning model was like overtrained to like one machine, and if you tried to do the same analysis on a different machine using that training data?
So we have two machines. We use different machines, but you are onto something, right? So for instance, when we deposit the droplets, we don't put the test groups always in the same side of the table. We have to basically move them around, because if we don't randomize that, then we do see effects from that as well. So it's you know, but those are things that we worked out and we realized we had to do. So this is, I think, goes along the lines of what you're saying.
That's a wonderful opportunity.
Yeah.
Graduate student.
Okay, yeah. But it's a good point, you know, and so with machines, I haven't systematically looked at it. I doubt that that's the case, but for instance, also the other thing is, if you don't make the surface right, you'll find difference. You basically measure surface difference in the surface as well. So that's possible as well. If you don't align them right, obviously the network starts looking outside of the droplets. So yeah.
So the 3D in vitro environment.
Yeah.
What types of experiments do you imagine those being used for in the future?
That's a good question. So I can tell you what we're working on, but I'm not sure if it's limited to that. But so we have a project with Eva Feldman where we're looking at essentially taking some of the IPS cells, patient-derived IPS cells, and then basically seeing if we can use this to derive brain organoids essentially out of those systems. And so we have, with healthy IPS cells, we've seen some success with this. And now we're looking at some of disease specific, because they have a lot of that. And there's an issue, how do you create essentially functional organoids out of that? And so we're evaluating and trying to understand if there's a benefit for this kind of systems in the context of disease models for ALS or Parkinson or something like that.
Yeah. So I was just wondering about those. I mean, are you able to get astrocytes and glial cells and neurons to play together? And
We do get a nice distribution of different cells, and we have analyzed that again with RNA-seq.
That's really neat.
And yeah.
That's very exciting.
And then sorry, just to follow up. Then the other area we're interested in is cardiomyocytes, IPS to cardiomyocytes, and there the alignment I think is going to be important. So because you want to get synchronization of the action potential, right, as it propagates over long areas, and you know, it works actually pretty well in monolayer cell structures, because the fact that they're all in one layer, in one plane, helps with synchronization. So when you go 3D, there's a huge challenge there to be better than what's out there in these monolayers. Right? Everybody says, "Oh, monolayers don't really," but when you actually work against them, it's not so easy. And so I think with the alignment of the ECM, potentially we hope we have some benefits there as well.
So, Jeorg, very interesting to hear about the third part. So the droplet drying is kind of a aggregation or phase transition process at increasing concentration.
Right.
The growing.
And there's salt there too. So basically there's a co-crystallization of the salt with the proteins.
It's like a free monomer and then grow into a polymer and for a certain shape, right?
So it's complex, right? So you basically get local saturation effects.
Yeah.
You get nucleation, then you get crystal growth, and then you get these interactions of the protein with the salt, where the protein can actually control the growth direction of the salt to some extent.
Right. But in the end, you did allude to how to build on it to do two-way interactions, a molecule A and B in that droplet, see how they grow together.
I mean, that would be good. We're still, the proof is still in the pudding. We're still trying to see if it's possible, but I think that would be really interesting. Yeah.
Right. So maybe AA aggregate early, then AB, then BB last, or something like that.
Well, or maybe you just see, if they are interactive, then they form a certain structure. If they're non-interactive, they form another structure.
Right.
If you get co-precipitation, maybe that's different than having one complex precipitating out.
Right. And then the advantage is you can do it with very high throughput.
Right.
Yeah, I see. I'll switch to a different question, which in the first part, what is the nature of the tissue specificity of those nanoparticles?
So certain proteins, albumin likes to go to the liver, obviously our particles do anyway, so that's not a good system, but the proteins themselves have tropism sometimes, right? Oh, you mean in the case of the
liver versus
you mean the sepsis model with Musikanter? Is that what you mean?
Basically, how it is serendipitously obtained specificity, or you can engineer to be super specific.
Yeah, so we think by picking the right protein, we can get tropism potentially. In the case of the sepsis model, I think I don't understand how it really works, but we think that neutrophils are the ones that basically are upregulated in the sepsis case, and then basically like those protein nanoparticles, which they probably have scavenger receptors for proteins, and then they basically selectively take them up in the case of sepsis.
Right. How would that compare with this other line of targeting something like adenoviral engineered expression that are tissue specific and then release only there?
I mean, if they had good proteins, we could learn from it. That would be good, right? So I mean, I would be, I would think if there are lessons to learn, and I just talked to David Schaffer. I don't know how much he wants to talk about it and so on, but I think there's not a lot right now in literature. But if we would learn about how they actually interact with what makes a tropism there, I think we could put some of that. We could build protein particles from those proteins. I think that's one way to do it. And then the other thing, obviously, is I mean, adenoviruses have their own issues with basically being used clinically. And so if we can essentially develop a synthetic system that doesn't bring the negatives of that, potential immunogenicity and so on, then that would certainly be not a disadvantage.
Right. I find this really thought-provoking because others have tried to engineer this delivery vessel with multiple layers of zip code, like one go to cross blood-brain barrier, another go to cell type, another go to intracellular organelle. So your nanoparticle can be one among the many layers for that address. Right?
Yeah, and maybe that's the solution in the end, right? Maybe maybe you make it bicompartmental. Maybe use the two compartments to actually co-present some of those targeting units.
Especially for physical property, like outside and inside have different pH or osmolarity. So nanoparticles would be good.
And we obviously need to deal with that too. So I didn't talk about this, but endosomal escape is something we think about a lot. And so there are components in there that help us to get out of the endosome, because we need to ultimately get into the cytosol, or when we do plasmids, we have to go into the nucleus, right?
Great, thank you.
Yeah.
Anybody online? Questions?
I don't see any online, but you're welcome to unmute if they would like to.
Any other questions? Oh.
I was wondering what factors control the release of the drugs from the nanoparticles, and would there be a situation where you could have the independent compartments releasing at different times?
Yeah, and we actually, that's a great idea. And so we have done that in vitro. So we, for instance, breast cancer, we've taken two different breast cancer drugs. That was work with Samir Mitragatre from Harvard together. So they did the cell experiments, we made the particles. And then basically ask questions about what happens if we release drug A first versus drug B first. And then we looked at the regime too. So we actually control that pretty precisely. And we saw actually very big differences in the combination index, depending on which you release first and what the sequence of what the periodicity of the release is. So I think that's very relevant and absolutely possible because we can control the matrix, right? We engineer the matrix in which the drug is embedded. And by doing so, we can control the release.
So I have a small comment, Joerg. I don't know that much about machine learning. There's a technique in numerical analysis called SVD.
SVD.
SVD, singular value decomposition.
Okay.
So the people use always like eigenfaces. Like SVD is like eigenvalue. You can use to get like called singular values and singular vectors. So there's an area called eigenfaces. So in my mind, like eigen droplets. You can create eigen droplets.
Hey, let's do that together.
Yeah, that's a word.
Yeah, as a word.
No. You can use that same principle here. Then you
Okay.
can invent a word eigen droplets.
Okay.
You see that DARPA as well.
Thank you, Joerg. Thank you
Okay.
so much.
Thanks a lot.
Thank you.
Well, thank you for having me. It was fun. Thank you.